So, in leading up to talking about diabetes mellitus, we really need to discuss the maintenance of plasma glucose because it's not something that we've had a chance to talk about in normal physiology. As we'll discuss more in the diabetes lecture, diabetes mellitus is a group of disorders that affect glucose homeostasis. And there's several different types of diabetes mellitus, and there's several different alterations in that maintenance of glucose homeostasis. But all of the forms of diabetes mellitus basically are syndromes that are characterized by chronic hyperglycemia and other disturbances in carbohydrate and fat metabolism. So we really need to understand the maintenance of normal plasma glucose before we can really discuss the alterations or the abnormalities that occur in diabetes mellitus. So normal blood glucose is maintained through the tight regulation of three basic processes. The production of glucose by the liver, and to some extent also by the kidneys, and that process is called gluconeogenesis. So in that process, energy substrate like lactic acid, like amino acids, are sent to the liver and the kidneys, and the liver and the kidneys take those substances and produce glucose. And that glucose is then put back into the bloodstream in order to maintain normal plasma glucose levels. Glucose is also stored intracellularly, and it's stored in the form of glycogen. And the primary places where we see stored glycogen in significant amounts is in skeletal muscle, and primarily that glycogen is for energy to drive skeletal muscle contraction. So the skeletal muscle has its own supply of glycogen to support its own contraction. And then we also have glycogen stores, significant glycogen stores in the liver. And the liver glycogen stores are designed to be a storage of blood glucose, basically. So glucose in the liver stored in the form of glycogen can be broken down and then released into the bloodstream when needed to maintain blood glucose. And then we have glucose uptake by peripheral tissue, especially skeletal muscle and fat and the liver. It's not on this list, but the liver is also an important part of that list of peripheral tissues. And this uptake of glucose by these tissue types is insulin mediated, meaning that the insulin allows these tissue types to take up glucose. Now, these different tissue types are specified, or the glucose uptake is regulated in these tissues because these are tissues that can derive energy by other means, particularly for the breakdown of adipose tissue, or the breakdown of fat. So the uptake of glucose by these peripheral tissues are regulated so that in times of low blood glucose, it can be limited. The uptake by this peripheral tissue can be limited, so that blood glucose can be saved for tissues that can't use fat as a source of energy, primarily nervous tissue, especially the central nervous system. So this maintenance of blood glucose is very important because we need to have blood glucose within a healthy range. If blood glucose levels were to get too high, hyperglycemic conditions are toxic for many, many tissue types. Even small, even moderate increases in blood glucose sustained over time really is very toxic for a variety of tissue types, as we'll discuss later. But then on the other spectrum, at the other end of the spectrum, we don't want blood glucose to get too low. We need an adequate level of plasma glucose in order to support brain function. So central nervous system function starts to get compromised when blood glucose gets too low. So blood glucose becomes toxic at very high levels, and it doesn't support adequate brain function at levels that are too low. So we need to maintain blood glucose at all times within a healthy range. But we also need to maintain this healthy range in the face of expected and normal significant fluctuations in blood glucose. And the fluctuations that are expected and normal occur around times of feeding and times in between feeding. So these two states that represent normal times of glucose fluctuation, we call them, number one, the absorptive state. This is the state where we are actually actively absorbing the food that we consume. So during the absorptive state, the priority is to decrease plasma glucose, or in other words, to prevent a marked elevation in blood glucose. And then the other state is called the post-absorptive state, also sometimes referred to as the fasting state. In this state, the priority is to maintain blood glucose, and if it happens to fall, to bring it back up. And so this state is, we are not consuming food, we're not actively absorbing glucose into the bloodstream through the GI tract. So we need to maintain the blood glucose that's there, and as the brain consumes more and more blood glucose, we need to have a way to replace it in order to prevent a marked drop. Okay? So we have these points where during and after meals, where we need to do something with the glucose we're absorbing, okay, so that it doesn't cause a marked elevation in blood glucose during the absorptive state. And then when it's been some time since the last meal, we need a way to maintain our blood glucose levels, even though tissues like the brain are constantly drawing glucose out of the circulatory system. Okay. So what happens during the absorptive state to help achieve this goal of maintaining a normal range of blood glucose? So under absorptive state conditions, we're consuming food, and that food, we are digesting it, and we are absorbing glucose through the GI tract, and so blood glucose levels are starting to rise. Now, the way that our body deals with this, the way that glucose is maintained, glucose homeostasis is maintained during an absorptive state, is that blood glucose, many things are done with blood glucose. Basically, we allow tissue to take up blood glucose, and then that glucose is largely stored in some form. So blood glucose and the amino acids that we are consuming and absorbing with our meal are taken up by skeletal muscle. The amino acids are stored in the form of proteins, and the glucose that's taken up is stored in the form of intracellular glycogen stores. The liver takes up blood glucose and stores it primarily in the form of glycogen, but also in the form of triglycerides. So glucose in the liver can be converted into triglycerides and stored within the liver that way as well. And then adipose tissue also takes up blood glucose. Also, it takes up any fats that we are consuming with our meal and absorbing. And then within the adipose tissue, the triglycerides are stored, and the blood glucose that's taken up is also converted into triglycerides and stored. Okay, so that's what happens during the absorptive state. In the post-absorptive state, the liver is going to play a key role in maintaining blood glucose in the post-absorptive state. And that's an important thing to remember. The liver has many, many functions in our body. One of its functions is to play a very key role in glucose homeostasis, and that's especially obvious in that post-absorptive state. So in the post-absorptive state, we're talking about conditions where it's been several hours since we've consumed food. So we're really in a fasting state. Um, and so we're not consuming food. Glucose is not being absorbed, okay? But it is being taken up by constantly by the brain, by the central nervous system, okay? But in order to spare the glucose for the brain, okay, in order to spare blood glucose for the brain, the uptake of glucose by tissue like the skeletal muscle, like fat, like the liver, is blocked. Okay? So these tissue types, this peripheral tissue, does not take up blood glucose in the post-absorptive state. Now, what happens as an alternative to that, or alternatively what happens, is that the liver does two things during the post-absorptive state to help support blood glucose levels. The liver is going to break down its glycogen stores and release that glucose into the blood. And it's also, if need be, going to take up amino acids in circulation, lactic acid in circulation, and other forms of energy substrate. And it's going to synthesize glucose, okay, and then put that into the blood. So the liver is going to break down glycogen, and it's also going to do this gluconeogenesis, this production of new glucose. Now, in a post-absorptive state, the liver is going to do much more glycogen breakdown, okay, than it is going to do gluconeogenesis. Okay, so your body depends much more on glycogen stores in the liver than gluconeogenesis in order to support blood glucose during a post-absorptive state. And then alternatively, this tissue that is not taking up blood glucose, skeletal muscle, adipose tissue, liver, this tissue alternatively are going to use fats as energy. So adipose tissue and circulating triglycerides are going to be broken into free fatty acids so that those free fatty acids can be taken up by tissues and used for energy instead of blood glucose. So strategies that are put in place during meals, during the absorptive state, include that we are going to shut off certain processes that we had going on during the post-absorptive state, and we're going to turn on other processes that we need for the absorptive state. So two of the processes that we shut off that we were using in the post-absorptive state is gluconeogenesis and glycogenolysis. Okay, so we see a decrease in those processes. We start to turn on glucose storage. Okay, so the tissue types, the skeletal muscle, fat, and liver, starts to take up glucose, okay, and it's going to store it in the form of glycogen, or in the case of fat, store it in the form of triglycerides. And also, these tissue types are going to start to limit the use of fat as a primary energy source and increase fat storage. So as the glucose gets taken up by adipose tissue, by the liver, it starts to be converted into fat and stored, okay? And we call that lipogenesis. So then during the post-absorptive state, okay, we are going to start to limit the access to glucose to only those tissues that absolutely need it, like the central nervous system. Okay. The liver is going to start to break down its glycogen stores, and that process is called glycogenolysis. It's going to start making new glucose. That process is called gluconeogenesis. And these tissue types that can do this, like the liver, skeletal muscle, and others, they are going to use fat as a primary energy source rather than glucose. So the breakdown of fat we call lipolysis. Okay. So as you can see, in the absorptive state, we are doing the opposite set of strategies that we do during the post-absorptive state. And this allows us to maintain glucose levels at a normal range despite these very, very different conditions. So in order to fully understand this topic of maintaining blood glucose and also leading up to understanding diabetes mellitus, we have to understand how glucose gets in and out of cells, because the uptake of glucose by cells is a very, very big factor in maintaining blood glucose, and then it also becomes a very big factor in the pathogenesis of diabetes mellitus. So glucose gets in and out of cells through a facilitated diffusion mechanism. We have glucose transporters. They are membrane proteins that carry glucose across the plasma membrane. But the glucose is moving passively. It's always moving down a concentration gradient, but these transporters are simply creating the mechanism for that, or the opportunity for glucose to move across. So it binds to glucose and it carries glucose across. So there are 4 different subtypes of the glucose transporter, and 2 of those subtypes are of importance to us. The GLUT1 transporter, or the subtype 1 transporter, is found on nearly every cell in our body. It allows glucose to enter cells. It is what we call constitutively expressed by all cells. And when we say constitutively expressed, it means that these transporters are always produced and they're always found on the membrane. And these GLUT1 transporters are found in low concentrations or low density of them on the membrane. And they are responsible for allowing a low level of just baseline glucose uptake that is required to sustain energy, basic energy levels by all cells. Okay. So it's found on all cells, not in huge numbers, in relatively low numbers, but it allows for just a baseline trickle of glucose into cells, and that baseline trickle helps maintain really basic cell function. Okay. Now, GLUT1 transporters, the expression of GLUT1 transporters can be increased or decreased depending on prolonged conditions. In fact, GLUT1 expression is increased with prolonged fasting and decreased with persistent exposure to excessive glucose. So there is some modification of the expression of GLUT1 transporters under significant conditions, but in general it's something that's found on all cells that just allows for a very, very small amount, basal amount of glucose to enter all cells. Now, GLUT4 transporters, on the other hand, these are the transporters that are expressed on muscle tissue, especially cardiac and skeletal muscle. It's found on adipose tissue. It's found on the liver as well. And GLUT4 transporters are responsible for the kind of glucose uptake that is insulin mediated. Okay. And so these GLUT4 transporters are not constitutively expressed. In fact, the availability of these GLUT4 transporters are entirely dependent on insulin. And the process looks something like this. So this is obviously a very, very simple cartoon representation of the interaction between insulin and the GLUT4 transporters. So number one thing to notice is that these GLUT4 transporters are found inside of the cell on the membrane of intracellular vesicles. Okay, so these GLUT4 transporters are found on the plasma membrane of intracellular vesicles. And this cell could be a cardiac muscle cell, a skeletal muscle cell, an adipose cell, and an adipocyte, or a hepatocyte, or a liver cell. When insulin binds to the insulin receptor on these cell types, it signals through a signaling pathway, a second messenger system, that we call the PI3K, or the phosphatidylinositol 3 kinase pathway, that stimulates, among other things, stimulates the exocytosis of these GLUT4 vesicles into the plasma membrane of the cell. So that means that these vesicles of GLUT4 transporters fuse with the plasma membrane, and that allows these GLUT4 transporters to insert themselves into the membrane, and glucose then can get taken up. Okay. So this is the mechanism by which insulin, it acts through the second messenger system to allow for the insertion of the GLUT4 transporters into the plasma membrane of these cells. Now, interestingly, when insulin binds to the insulin receptor, it does many things to the cell, creating the insertion, or allowing for the insertion, of the GLUT4 transporters is just one of the things that insulin does to these insulin-sensitive cells. In fact, so here we have the one arm where insulin binding to the insulin receptor, acting through this PI3K signaling pathway, causes the GLUT4 transporter to get inserted, glucose uptake. Okay. There are many other intracellular events that are stimulated by the PI3K signaling pathway. It stimulates proliferation, it stimulates the synthesis of lipids, proteins, and glycogen, which makes sense. The cell is taking up glucose. So the insulin is also stimulating all of the enzymes necessary to convert glucose into triglycerides and store the cell that can then store the glucose in the form of triglycerides. Insulin is also stimulating all the enzymes that are needed to synthesize glycogen. Okay, so this glucose that's getting taken up can be stored in the form of glycogen. It also stimulates through this PI3K pathway, and also through another pathway called the MAP kinase, or the MAPK pathway, also stimulates proliferation and cell growth. So the glucose that's taken up by this cell can also be utilized, okay, as an energy source to be directed towards cell growth, cell proliferation. Okay. So insulin, in addition to allowing for the glucose uptake by these cells, also stimulates all the enzymes needed to store the glucose, and it also stimulates many of the intracellular processes that are going to consume glucose and allow this cell to proliferate and grow. Okay. So insulin, we think of it often as primarily being involved with glucose uptake, but insulin also in many ways acts as a growth factor, and that's something to really keep in mind. So glucose homeostasis, as I mentioned, is maintained through these two very different conditions, the absorptive state and the post-absorptive state. And we have two hormones that are critical for each of these states. Now, insulin, I've already introduced, is one of them, and there's another one. Now, both of these hormones of glucose homeostasis are produced by the pancreas, and the pancreas is an organ that has both exocrine and endocrine components to it. The pancreas is depicted here. It's just below the stomach, very close to the duodenum. And you can see the exocrine portion of the pancreas where the pancreatic ducts that produce the sodium bicarbonate solution and produce all of the digestive enzymes that then get mixed with bile and are secreted into the duodenum and are absolutely critical for food digestion. So the pancreas is absolutely critical for food digestion, the exocrine portion. And then the endocrine portion of the pancreas is organized into these islands of cells that we call the islets of Langerhans. Okay? And if we take a closer look, what we see here is a portion of pancreas. It's a cartoon representation of a portion of pancreas. We see the ductal portion, the exocrine portion in yellow, and then at the center we see one of these islets of Langerhans, and it's composed of multiple cell types. Two of these cell types are of particular interest to us, the alpha cells and the beta cells. Now, the alpha cells and beta cells produce two different hormones. The beta cells produce insulin. The alpha cells produce a hormone called glucagon. Okay. Now, these are the two hormones that are critical for the absorptive state versus the post-absorptive state. Insulin is the hormone of the absorptive state. Insulin is the hormone that inhibits all of the enzymes for gluconeogenesis and glycogenolysis, so shuts off those two processes. Insulin allows for the insertion of those GLUT4 transporters and therefore increases the permeability to glucose of these tissue types, the skeletal muscle, fat, and liver. Insulin is what increases the enzymes necessary for glycogen synthesis, and it also increases the enzymes necessary for lipogenesis and inhibits the enzymes for fat breakdown. Okay. And in fact, any amount of insulin is has a very strong maintenance signal for fat tissue. Okay. So any concentration of insulin in the bloodstream is any significant concentration of insulin in the bloodstream is a strong signal to fat cells to maintain their density. Okay. And so the opposite is true. When insulin levels become very, very low for prolonged periods of time, that's a very strong signal for fat cells to start to significantly break down. Okay, and that becomes very important as we get into the diabetes mellitus section, as I'll explain when we talk about the different types of diabetes mellitus and how it causes how it affects fat cells. And then glucagon is the hormone of the fasting state or the post-absorptive state. Glucagon is the hormone that stimulates the enzymes for glycogenolysis. Glucagon stimulates the enzymes needed for gluconeogenesis. The presence of glucagon and the drop in insulin is what causes these GLUT4 receptors to get retracted from the membrane, and so skeletal muscle, fat, and the liver can't take up glucose anymore. Glucagon stimulates the enzymes necessary for the breakdown of fat, lipolysis, and it inhibits the enzymes necessary for fat formation or lipogenesis. Okay. So the actions of insulin, actions of glucagon, absolutely antagonistic to one another, so they are absolutely opposite. Insulin and glucagon are our classic example of antagonistic hormones. They are what we call reciprocally regulated and released, meaning that the presence of one hormone strongly inhibits the presence of the other. And these two hormones have completely opposite sources of stimulation. So the stimuli for insulin release is the opposite stimuli for glucagon release. When glucose levels rise, insulin is stimulated. When glucose levels drop, glucagon is stimulated. So opposite sources of stimulation. And when insulin is present, when insulin is released, that is a strong inhibitory signal for glucagon secretion. And when insulin levels drop, that in and of itself is a very strong stimulation for glucagon release. Okay. So very, very important relationship to keep in mind.